Hello, my name is Ed Chapman, and this is our third video cast for photosynthesis. And we stopped part 2 talking about non-cyclic photophosphorylation, which, if you remember, is the process of adding a phosphate group to ADP to change it into ATP using energy from sunlight or energy from photosynthesis. That's why it's called photophosphorylation. And this is the process that the electron transport chain accomplishes, and it's done by chemiosmosis. If you remember, the pumping of hydrogen ions from one place to another and then allowing them to backflow, and in the process of backflowing generate ATP using the protein ATP synthase, which is bound in the electron transport chain, part of the membrane. And it's non-cyclic because the electrons coming out of photosystem 2 are replaced by electrons from water, and the electrons leaving photosystem 2 and traveling through the ETC to photosystem 1 are then bound and eventually captured by NADPH to make NADPH, which is our electron carrier in this process. So it's non-cyclic in the sense that the electrons don't recycle. They don't go around and around in a loop. They travel in a chain. It makes ATP using photosystems 1 and 2. It needs light energy of the correct wavelengths for this to happen, and it replaces electrons taken from the chlorophylls with electrons from water. Here's a demonstration of the exact same process using little guys and hammers instead of light. So the light energy is represented by the swing of this mallet, which bumps an electron up. Okay, that electron, remember, is coming from photosystem 2, which is getting its electrons by splitting water. These electrons then roll downhill and turn a chemiosmotic wheel, so to speak, to generate some ATP. Then they get bounced uphill again and finally end up in the NADPH bucket, which is the photosystem 1, which is the NADPH producing photosystem. Hope that helps. I think it's kind of a cute diagram. Now, electrons can also flow in a cycle in the photosystem, and this doesn't produce any NADPH, but it does make ATP. So it's kind of like a short circuit. So the electrons are bouncing out of photosystem 1 and then falling back into photosystem 1, and in the process making some ATP. Now this cyclic electron flow is important because ATP is needed. There's more ATP is needed than NADPH by the Calvin cycle, which we're going to talk about later. And because the system needs more ATP than NADPH, this is a way for photosynthesis to make a lot of ATP without producing too much NADPH. And here it is. The Calvin cycle consumes more ATP than NADPH. Therefore, cyclic flow makes up the difference. It makes up for the difference of needing more ATP than NADPH. All right, now, to review what we've been talking about here, all this is involving electron transport chains embedded in membranes. And we did a little diagram of this in class where we labeled a worksheet. And as you guys can see here, chemiosmosis is the process of using the flow of electrons to pump hydrogen ions from one place and allow them to backflow to where they came from, and in the process power an enzyme called ATP synthase, which then phosphorylates ADP and changing it into ATP. All right, now in a mitochondrion, this pumping is done from the matrix out into the intermembrane space. Okay, so it goes from the inside of the mitochondrion outward, and then the hydrogen ions flow back in and generate electricity. Generate ATP. In a chloroplast, it's just the opposite. Okay, in a chloroplast, the hydrogen ions are being pumped from the stroma into the thylakoid space, and then they're flowing back into the stroma to generate ATP by chemiosmosis. So in a chloroplast, the electrons are being pumped from the stroma inward into the thylakoid space, and then they're flowing back out in chemiosmosis. Just the opposite direction that we saw in a mitochondrion. So in a lot of ways, mitochondria and chloroplasts kind of do the movement of electrons backwards. Mitochondria allow them to fall downhill to make ATP, exergonic reaction. Chloroplasts pump the electrons uphill in the first few steps of the photosystem to put energy into them, and then allow them to fall downhill to make ATP. All right, so let's compare the chemiosmosis of respiration with the chemiosmosis of photosynthesis. So where does it happen? Okay, and what is the direction of hydrogen ion pumping? In the mitochondrion, in the process of cellular respiration, it takes place on the inner membrane of the mitochondrion. In the chloroplast, it takes place in the thylakoid membrane. And the direction of pumping in mitochondria, in respiration, it's from the matrix to the intermembrane space, which is pumping in the outward direction. And in chloroplasts, it's from the stroma into the thylakoid space. So that's an inward direction. So, like I said earlier, it's they're kind of the reverse of each other. Now, the light-independent reactions are the part of photosynthesis that doesn't require light to happen. So these reactions can take place in complete darkness as well as during the day when the sun is shining. They're also known as the Calvin cycle, named after the guy that put it together back in, I believe, 1958, if I remember right. It's also known as carbon fixation because the light-independent reactions take carbon dioxide from the atmosphere, which is floating around free, and lock it in place in carbohydrate, which is a solid, something you can store. The light-independent reactions, otherwise known as the Calvin cycle, require ATP and NADPH to run and make carbohydrate. And as I said, they can happen day or night inside of plant cells. Similar to the Krebs cycle, but different in some important ways. Okay, and if you remember the Calvin cycle on the left, which is our new photosynthetic cycle, and the Krebs cycle is our old one, which we did during respiration. So the Calvin cycle removes CO2 from the atmosphere and fixes it into solids we call carbohydrates or simple sugars. The Krebs cycle does just the opposite. It produces free carbon in the form of carbon dioxide. The Calvin cycle is powered by ATP and NADPH, and the Krebs cycle is powered by ATP and NADH. A slight difference there. The Calvin cycle only takes place inside of plants that have photosynthetic pigments, and the Krebs cycle only takes place in both animals and plants. All right. The Calvin cycle happens in the stroma of chloroplasts, and the Krebs cycle happens in the matrix of mitochondria. Okay, the Calvin cycle up close. So let's look at what this wheel, or this turning wheel of chemical reactions, is actually doing. And if you first look at this, we're putting in carbon dioxide, and we're spitting out small sugars, small 3-carbon chains, which 2 of them put together can build a glucose. So that's our output, and carbon dioxide is our input. All right? Now, there's an enzyme right here at the first step called Rubisco, and it's the enzyme that actually catalyzes or causes to happen at a faster rate the fixation of carbon dioxide in the first step of the Calvin cycle. So Rubisco is interacting with carbon dioxide to fix it and lock it in place to produce this chemical here, which is 3-phosphoglycerate. Okay, so Rubisco is important. Rubisco is probably the most common protein on Earth. All green plants use it, and it's the enzyme, the protein enzyme that is fixing carbon dioxide in the air eventually into carbohydrate. So for every 3 carbon dioxide molecules that are fixed, we get 1 3-carbon carbohydrate produced. So 3 carbons in, bonding with Rubisco to make this chemical, and then this chemical, and then finally this chemical, and out comes part of that as a sugar. The rest of it continues around to regenerate what you need for Rubisco to grab onto in the first step. So it's an endless cycle that's spitting out carbohydrates. Now, plants have a problem in hot, dry climates, and the hotness, the low humidity, makes it difficult for plants to fix carbon dioxide, and you need to understand how this works. We're going to do this, though, in the next part of our photosynthesis video casts. I'll save that for next.